When we come across something like type 2 diabetes, obesity, metabolic syndrome, super complicated processes, we're still looking for that magic bullet, that single nutrient, that single medicine, whatever it is. And that's why doctors particularly, are really well trained on pharmaceutical drugs. They're really badly trained on lifestyle and trying to work out complex solutions to problems.
Welcome to the Proof Podcast, a space for science-based conversation exploring the health and longevity benefits that come with mastering nutrition, physical exercise, mindfulness, recovery, sleep, and alignment. Facts, nuance, and trustworthy recommendations, minus the hyperbole. Hi friends, great to be here with you. I'm your host, Simon Hill. I'm a qualified physiotherapist and nutritionist with an undergraduate science degree and a master's in the science of human nutrition. Today I sit down with Dr. Tim Spector. Dr. Spector is a medically qualified professor of epidemiology and director of the TwinsUK Registry at King's College London. His current work focuses on the microbiome and nutrition, and he is co-founder of the company Zoe, which has commercialized a home kit for personalised nutrition. In this conversation, we focus on his research on twins, how two people can be genetically identical but experience very different health, what makes us most unique, and how personalised nutrition may be the key to unlocking the healthiest versions of ourselves. Hey Tim, welcome to the show. I appreciate you taking the time out of your day to talk all things nutrition with us today.
It's a great pleasure.
There's a lot that I want to talk to you about, and we were just chatting off air. I think this will be hopefully one of several conversations that we have. I always find your posts online and your journal articles, of which there are many. How many journal articles have you published so far?
It must be getting towards 1,000 by now, but I don't count them every day. So
right.
It's very high numbers, embarrassingly high actually. People say, well, you can't possibly have written them all in great detail.
Well, there's a lot, and of course your books, one of which I have in front of me here, Spoon Fed. I've always really enjoyed your books. I find them extremely insightful. Given that it's your first time on the show, Tim, it would probably be nice to kind of start here by hearing from you how this very, very big, successful career of yours in science and nutrition science came about, and how it's sort of evolved over the last few decades for you.
It's an interesting question, really, because I think it's been an evolution rather than things suddenly happening. And I started as doing medicine in London and was much more interested in the research side of it, but got drawn to clinical medicine and did get interested in epidemiology as a medical student, and actually wrote a paper as a medical student, which got me sort of interested in that field. I then went on and did rheumatology, so as a physician, which is arthritis research and arthritis, bones and joints. Became a consultant, which sort of gets to the top of that tree. But on the way, I stopped and did a master's in epidemiology, and really did this sort of epidemiology of bone and joint diseases. My thesis was in hormones, menopause, and arthritis. And then in about 1993, needed a break from my supervisor and a new career. And that's when I decided to start the Twins project, which was having the biggest twin registry in the UK, and started working on that full time. And that was a real big success. And we ended up with 15,000 twins. And then I started branching away from just doing arthritis and osteoporosis, and started really studying every disease under the sun, both physical and mental and psychological, using the same methods of this nature v nurture idea of twins. And that was amazingly successful because no one else had that resource, and able to answer all kinds of fascinating questions. particularly at a time when people were thinking all these chronic diseases were just wear and tear. Oh, that's just old age, and the idea that there were genetic components to it was totally new. Mm-hmm. Discovering things like back pain is actually 3 times more genetic than breast cancer. It was that kind of stuff that we were bringing out. And then obviously discovering genes on the way. And I sort of taught myself genetics and genetic epidemiology and sort of became an expert after a while. And then I started getting interested in why twins were different. Why identical twins didn't always get the same diseases. They didn't always have the same personalities. One would be fat, one would be thin. One would be depressed, one would be happy, one would get cancer, one wouldn't. So then I started saying, well, should be the perfect model to tease out when you've got 2 genetic clones, why one's different to the other. So then I got interested in epigenetics. And I thought that was going to be the answer. This is the study of how chemicals can switch on your genes on and off. And so, your genes aren't this absolutely fixed item. You can slightly tweak them. And that's why identical twins could be different. And that led me a certain way, but I found it wasn't really a big change. And it was really hard technologically to follow that up. The science wasn't quite there. And so that then got me onto looking for something else, and that really got me onto the microbiome. And I think that was the turning point for me when I suddenly said, this is about 12 years ago, getting into the gut microbes, discovering those guys, they were so different that said, this is my field now. And this suddenly makes nutrition interesting, from being the most boring science in the world to being the most exciting science. And that's where I made this switch again in my career to really move from genetics, epigenetics, into nutrition and the gut microbiome.
So one of the things that I kind of want to untangle in this conversation is throughout your career, how your views of nutrition and how food and diet relates to health has changed, how it has sort of evolved over time. Back in your days working as a rheumatologist, and at that time, do you recall what your views on nutrition were, and did you have sort of any strong views with regards to how food affected health at that time?
I think I had the very traditional views of a doctor who was brought up having had, maybe 3 or 4 hours of nutrition training in the sort of 5 or 6 years of medical school. So very limited. We were taught much more about scurvy than obesity, for example. Mm-hmm. And still medical students still are, although we never see a case of scurvy. It was really that, obesity was this thing that just happened. It was more about willpower. People were just lying about what they ate, and that you were just told, tell your patients eat less and move more. That's the only real way, and there were no overweight people in concentration camps was a sort of mentality that was rather brutal at the time when you were seeing these very overweight patients. That's what my bosses were saying to the patients. So it was very unsympathetic approach, and it was we really had no answers either, because particularly in rheumatology, there are many overweight patients suffering with really bad arthritis, and the arthritis made the problem worse. They really couldn't move, and they were in pain, and the whole thing was a very bad vicious circle, and they ended up getting diabetes as well. So it was a time of great ignorance when you look back, and I'm not at all proud of myself and what my colleagues were doing, and but when we first started, there weren't that many morbidly obese patients. It was something that came on in my career. And I remember when I was working for a year in Belgium, in Brussels, and saw for the first time, actually, they had a speciality that had specialized in overweight patients in obesity. And that didn't exist in the UK at the time. There was nobody trained in how to deal with very overweight patients. So everyone just made it up as they went along. Mm-hmm. It was bad news. And diabetes was something, no one really, it was a very minor thing when I started, type 2 diabetes. And we learned very little about it in medical school. And it was just drug treatment was the only way forward. So it's changed massively as I've gone through medicine, really, as we've seen these epidemics come at us and totally ill-prepared for it.
Do you think that most of that can be attributed to the fact that you mentioned there scurvy, right? And sort of in the early 1900s, I imagine it made a lot of sense to focus on a single nutrient. And I know that you talk a lot about nutritionism. Is it applying the same lens? We tried to apply the same sort of lens to a different series of conditions. Is that what you're getting at in terms of not really having the right tools to combat these chronic diseases that were quickly developing?
I think so. Well, it's all part of this reductionism that is, medicine is not much different to other branches. We like to have a simple solution and work out, it's this deficiency in this chemical. If we give it back to people, we can cure them. And this was very much the teaching of the time. So that's why medical students today still learn about scurvy, because it's a lovely story that if you find out it was the vitamin C causing, it was the villain, everybody wins and everyone says how brilliant you are. And we keep searching for this these equivalents in medicine. And when we come across something like type 2 diabetes, obesity, metabolic syndrome, super complicated processes, we're still looking for that magic bullet, that single nutrient, that single medicine, whatever it is. And that's why doctors particularly are trained, they're really well trained on pharmaceutical drugs. They're really badly trained on lifestyle and complex, trying to work out complex solutions to problems. So I think that's really where we are. And that's why medicine does focus on these single chemical solutions. And they love the thing, this chemical in the brain does this, we can therefore switch it off with a drug. And that's this huge fallacy that was able to cope with disease in the past, in particular infections and that, but it's totally not suited for this complex array of complex diseases we're seeing in the modern era. And I think that's really what I was getting at, and how my training, was largely just disappointing for many of these diseases. Yet in small areas, it was amazingly successful.
You mentioned earlier that it was the twins research that sort of led you to start thinking about the microbiome. And you also just mentioned then that these conditions like obesity and type 2 diabetes really started to gather pace, particularly in the last 30 odd years. And I'm interested in this kind of discussion about genetics, epigenetics, and the microbiome. Would I be right that a condition like obesity is highly heritable? There are many obesity-related genes, and that humans, for, I guess, presumably thousands of years many more than that, have had these genes and had a susceptibility to storing excessive fat, but then it's taken a change in the environment in order for that to lead to the overweight or excessive fatness, adiposity that we see today?
Yes. I mean, most studies, if you take family studies, adoption studies, and twin studies, put the genetic component to obesity around 50 or 60%. So
right.
The heritable component, that is how much of that is explained by your genes, is certainly, is over 50% in most of these studies. So there is a susceptibility to it, and there's also we did studies showing that where you lay down that fat in your body is also largely genetic. So whether you get the beer belly or the big bum or whatever it is, that's your genes are sort of deciding where to store that fat in your body, as well as internal fat. So we know that's a big problem, what's called the visceral fat around your organs and your stomach and your liver. Again, there's a predisposition to that. And I think if you look evolutionary wise, this was generally beneficial in the past because we used to episodically run out of food, and therefore people that were really effective at storing fats did well. And so the idea of the genes was really to maintain this body weight at all costs. So you would it wouldn't really make sense to have an upper limit of it, and that's why we're in this problem now. So we've got a set point that cuts in that stops us burning energy when we get low, but there's no real upper point. And in the past, that hasn't really been a problem until we were faced with this overabundance of food, cheap food that was available, that was super tasty, that made you even more hungry when you ate more of it. It is combined with an environment where we're perhaps moving, less than we used to be. So I think, so the genes that served us well in the past are not serving us well at the moment. So we lay down a lot more fat than we would have done in the past because we're not having those periods of famine where it was useful. I think that's the way to see it. But it's quite clear now. There's a very good lower set point where our body does everything to keep our body weight at a certain point, and that's why people on starvation diets just plateau out. But at the top, there isn't that set point, and most people will just keep going on up. But it's interesting though. About a third of people just seem to have some resistance to getting very overweight, and that's another interesting part of it. That even in countries where there's massive obesity, you never find 100% of the population obese.
How many different genes are there that have been kind of identified that relate to obesity or would predispose someone to gaining excessive fat?
There's several hundred genes. It depends on how your threshold for deciding, what percent. Because the bigger your sample size, the more you can show tiny genes that have a minute effect of 0.01%. Sure. And once you get to 1 million people, you can start to show these effects, which are very big at a population level, but very trivial at a personal level. And I think this is the problem. So people say, yes, we've discovered, 500 genes for obesity. That means we can, understand obesity. But if you put them all together, they maybe only explain 1 or 2% of any one individual's genetic composition. So that even if you understood them, you wouldn't really be able to predict much about that person. And that's the big difference that's why genetics has been disappointing in the science of chronic diseases, because we've found thousands of genes, but sticking them all together, they only account for a very small percentage of the condition. So you can't use them as a personalized predictors of things that we thought we were going to.
Right, but if we're thinking on the individual level, we all kind of know someone who seems to not have too much trouble at all staying thin, and then someone else who is seeming to try to do all the right things but is really struggling. How much of that could be down to genetics versus other factors like the microbiome?
I think if you'd asked me 5 years ago, I'd have said most of that is genetics. And certainly, just the overall maintaining a weight had definitely has at least 50% genetic factor. But knowing that the way we respond to food is not nearly as genetic from our latest research, and that using twins to see when you actually do dynamic studies on twins, how does their body respond to the same food? It's much more different than we thought. I think the environmental component, the microbiome component, could be much more important than we've believed before. So I'm sure it's a mixture, but there's definitely some genetic component, because we all know families where everybody stays skinny and they can seem to overeat, and others, it's the opposite. But I think there's quite a lot of middle ground and interaction between the genes and the environment, and that don't depend on genetics. And because we know the microbiome is only very weakly heritable, estimates around 5% or something, that's this other whole organ that is outside this genetic control that I think is important. So it's going to be a bit of both. I'm not going to go and put a percentage on it, because I don't think we know enough about these complex interactions, and individuals might be different, and there might be, we can't assume that all the genes are working the same way. And there might be some families where these genes really are very strong, and others where, it really is the environment and the microbiome that's stronger. So I think we have to keep an open mind on it.
If someone's listening and thinking, what are these guys talking about, genes and obesity? Are we talking about various genes that could be affecting things like your appetite, hormones, so regulation of appetite, or are we talking about genes that regulate your sort of basal metabolic rate? How would these genes kind of be interacting with this body fatness that we're talking about?
Most of the genes that have been found so far, when you look at their function, that have been associated with obesity in these massive studies we're talking about, where you get, 250,000 people, and you look at their, you compare their BMI, body mass index, with their genes. Most of them tend to be involved in the brain, interestingly. So the genetic control of the brain, which is probably around appetite, satiety, fullness, your drive for food, or how quickly you feel the need to have more food again. Subtle changes there look as if from the number of genes that's more under genetic control than, say, these other metabolic rate mechanisms. Although we should say we still don't have very good methods of measuring metabolic rate or understanding how we burn calories. So it could be that we're missing a lot because we can't identify those. But so far, most people are quite surprised at how much many of these genes are brain function genes rather than adipose function genes. But whether we've scratching the surface or we really have got a full picture, I don't think anyone quite knows yet.
Something that's always interested me, and I'm sure you're familiar with this study, a colleague of yours, Christopher Gardner, the Diet Fit study, comparing the low carb with a high carb, sort of high quality diets. And I think they did look at about 3 different SNPs, genetic sort of variations and insulin resistance. You probably know this study better than I, but they were looking to see if they could predict who was going to do well on a low carb or a high carb diet. And they already from a prior study had the sort of prior knowledge, realizing that you probably won't see much difference in terms of weight loss between the two groups. And within each group, you'll see some people that do really well and some people that do poorly. And they showed that on a waterfall plot. And it wasn't explained by the 3 SNPs, and it wasn't explained by insulin resistance. And I've always sort of walked away from that study thinking, well, is it genes that we just aren't aware of, that weren't looked at? Or is this where the microbiome comes in? Sure, it could be behavioral things and it could be many other things that could explain this. But do you think potentially that the microbiome could dictate why someone may do better on a low carb diet versus, say, a higher carb diet? Because if we look at these studies that have been done comparing these diets all the time, and if you jump online, you will hear, many stories of people doing diets from a macronutrient point of view that are opposite and both seemingly, reporting benefits.
It's quite likely that is the case. And obviously, until you measure absolutely everything, you can't be sure what the component is, or you do some actual intervention studies. But I think if you put the Diet Fits study together with, say, the first of the Zoe Predict studies, where we were looking at interventions. So the Diet Fit study was comparing low-fat versus low-carb healthy diets and trying to predict who did better or worse. And they really didn't come up with any great prediction, other than there was huge variation between them. And the Predict study was giving everyone the same diet, which was basically these muffins, and see how they responded over the next 2 weeks. And the interesting thing was that I expected there to be quite a big genetic component. That's why we put in 600 twins into that study, thinking that the genetics would be quite key into predicting how an individual will respond to either fat or to sugar. And it was clear pretty quickly that genetics were not a major player, because they explained about 30% of the sugar responses and somewhere between 0 and 5% of the fat responses. So vast majority of our responses to food in fats or sugars are nothing to do with our genes. And if they're nothing to do with our genes, what else could it be? It's the microbiome is definitely the number one contender there for something that can change it, because we know that it is involved in the way we break down our food, we break down our fat, the bile salts, everything else. So it all starts to make sense then that that is the case. And and for me, having studied twins and genetics for 20 odd years, it was a bit of a surprise that genes weren't more involved in that part of the way we process food. I thought it would be that control. And maybe it comes back to your earlier question of saying, well, if the genes for obesity are more in the brain, then actually the way we sort of metabolize food and everything else is outside that genetic control. And there are these different processes going on rather than this. It's all one simple unified approach. So I think the microbiome definitely trumps genetics when it comes to how all of us process our food differently, and adds to that individuality, which I think is a really important fact.
And how much does that sort of dictate, I guess, epigenetics or microbiome environment dictate our health versus genetics? So if you look at these twins, let's say genes were fully responsible for our health. The genes we're born with dictated our entire journey through life. We would expect twins to have the same outcomes, right? Identical twins. Whereas you alluded to it earlier that we don't see that, and health outcomes between identical twins can in fact be different. How different? What are we talking about there?
When you look at the body shape, very similar. So waist to hip ratios, where the fat's distributed, you get, the shape of identical twins really looks very similar. So at that sort of physiological level, things are really quite similar. But where things start to go different, even in identical twins, is outcomes like having a heart attack, having cancer, so having an autoimmune disease. So just to give you some examples, rheumatoid arthritis, one of the commonest autoimmune diseases is if one twin has it, the other twin only has a 1 in 6 chance of also getting it. Mm-hmm. Whereas you would have thought that would be nearly 100%. These are genetic clones. They've lived their life together. They're brought up in exactly the same way. It should be more common than that. Cancers, about 1 in 5 chances, that kind of thing. So, and then when you get to longevity, actually there's very little correlation between when one twin dies and the other one dies. So it's a strange thing that you get all these the physiological similarities, the short term ones that are sort of wow, but the actual hard outcomes, strokes, heart disease, cancer, severe depression, hospitalizations, they seem to be more random events that are not explained by our genes. And that's what led me on this journey to try and see, was this epigenetic or was this due to things like the microbiome? And so that's what most people don't realize about twins, is that they are often more different than they look, because they're spookily similar when you look at them, but scratch the surface and you do find these quite big differences and these great anecdotes of extremely different twins, but personality as well as in their health.
It's an optimistic story when you kind of unravel it, because ultimately it means that the sort of code, the information that we're born with, while it does have some say in terms of our health, there is perhaps a lot that we can do, and hopefully, science has already uncovered a lot, but with years to come, we continue to uncover more and more, and that's just giving the individual much more control over their health, as opposed to some sort of predefined destiny. The other side of me thinks that if it was just purely controlled by genetics and there was nothing we can do, Twitter would probably be a much friendlier place. There'd be a lot less arguments. But it is what it is. Let's zoom in on the microbiome. Let's dig a little bit deeper there. So talk to me about the specific things that you found through your research that lead you to believe that quite a lot of our health outcome is modulated, affected by the composition of our microbiome.
It's a whole series of little pieces of information rather than one knockout blow. So I guess the first thing which we realized, even 10 years ago when we started looking at this with fairly crude microbiome analysis with what was called 16S analysis, where you just take one gene from all the microbes and you use that to work out what's going on, saw that in nearly every case where you said I've got 100 cases of a disease and you had 100 controls who were healthy, you always saw that there was less diversity in the case group. So for virtually any condition, the universal default position was that a disease group will have a less healthy looking gut microbiome than the controls. Even when you adjust for everything else, age and weight and sex and smoking and everything else you can think of, the disease seemed to have this effect. So there was this link between the microbes and disease. That didn't tell you about direction of causality, but it certainly said these are consistently associated in every single study. Then, of course, you've got all the animal studies where you could transfer the fecal samples to from one diseased animal to a sort of healthy animal and make it diseased, which made you think that they were having a causal effect. Well, it wasn't in humans, but it was a very nice, you could take human microbiomes and use these animal models to shift weight or cause inflammation, or you could cause anxiety or depression in mice, suggesting that there was a causal relationship of what was going on in the microbes to all this. Then you had obviously the successes of fecal transplants in humans, which haven't quite gone at the pace we thought they were going to go 10 years ago, but still there are a few successes. And I would say, the fact that you can get remission in something like ulcerative colitis in 1 in 5 cases by getting a donor sample put into someone with a colitis case and effectively curing that person. It doesn't work in 4 out of 5, but it does work in 1 out of 5. All these stories are telling you that the microbiome is playing a key role in health. And as we drill down into the individual microbes and we work out that they have properties like anti-inflammatory properties, that they're also, they can interfere with the medications you're taking and so make them effective or ineffective, shows that they are really playing a really active part in our health. And by changing them, you can also change your health. So I think it's an increasing list of studies that are being ticked off, showing how the microbiome, and particularly the chemicals they produce, are key for our health, because we know from many other work that some of the chemicals they do produce, like, for example, serotonin in the brain, is a really healthy chemical. So the idea they are these pharmacies is growing. So I think we also know that from our work that the microbiome plays a role in how you respond to food, and that it plays a role in these inflammatory sugar spikes, or how quickly you get rid of fat in your body. And, what we thought was only produced by the liver, these bile salts, is now also produced by your gut microbes. So really they've become a key organ in our bodies that we had no idea about really, even 10 years ago. Suddenly they are coming into every part of our this whole health. So yeah, it's a very long answer to a very short question, but I think it's a whole series of small bits of evidence that are accumulating to show why they are crucial factors in our health. And importantly, unlike most of the other organs in our body, much easier for us to manipulate and to modify if we can understand it.
So I want to go into some of the work that you're doing with Zoe and how you're investigating this to better understand it. I've had Dr. Will Bolsowitz on the show a few times, and the Sonnenberg. So we have spoken about the microbiome, but if someone's kind of catching this episode and hasn't listened to those, I do suggest they go back and listen to those. But at a sort of high level here, how does food actually interact with the microbiome? I think that's an important point for us to kind of just go over. And why might people respond differently to different foods based on their microbiome composition?
So at a high level, when you eat food, depending on the type of food, it may or may not reach your gut microbiome, is one thing to realize. So cheap, highly refined, ultra-processed food, or you're just having a sugary drink, for example, may go straight into the small intestine and the sugar gets absorbed into your bloodstream with very little interaction with your gut microbiome. We don't know that's absolutely true, but that's what we think. Other food that might be healthier, that contains, say, plants with high in fiber, will get further down into the gut, into the lower intestine, where that's where most of the microbes are. That's where they interact with it. And there will be several components of the food that will interact. And one of the key parts is the amount of fiber. Fiber is basically there, so it will be protected until it reaches the lower part of the intestine. And that's where the microbes will attack it and break it down into its constituent parts and extract all the nutrients and create these other great chemicals on the way. And they also extract from the plant something called polyphenols, which is the other important thing that we've only really recently realized, that are like rocket fuel for your gut microbes. So we can't process polyphenols directly, but our microbes can. And by using the fiber and the polyphenols, our microbes are then sort of energized to produce all kinds of chemicals, vitamins, everything else to control the rest of our body, control our immune system, etc. So that's the essential way. Then also you've got, you can break down sugars. The microbes break down fats for us. They're fat-eating microbes, just as there are fiber-eating microbes, and they break it down into these smaller components, these small fatty acids that get used, and protein as well. So in a way, we have a whole niche of little animals in there that are taking all the components, all the different foods, and the greater the variety of the food you're eating, the more this colony of little microbial animals will be, and the greater diversity of the chemicals you produce. That's my vision of this. And I think we're gaining more knowledge about how, each week, about how important the microbe processes are, the enzymes they produce are in breaking down that food and really doing it efficiently so that there's no waste and we're not hanging around lots of blobs of fat in our bloodstream, or we're getting excess sugar that's causing all kinds of stress to our body, and there's not so much inflammation. It's all dampened down. So all this depends on our microbes really functioning really well all the time.
Yeah, I don't often hear many people talking about protein and fats and the interaction between those and our gut bugs. And I saw a paper the other day. I haven't dug into it in detail, but it was looking at omega-3, DHA, and EPA, and I was surprised to see that they seem to find that they DHA and EPA, some of the fats at least, made their way down to the large intestine and had a prebiotic effect. So it'd be kind of interesting to watch that space of research as it evolves. The other half of my question, Tim, was what about our microbiome may dictate the types of food that we feel best on, that leave us feeling energized, vital, and that positively affect biomarkers of disease?
Yeah, well, it's a tricky question. So, I mean, in a way, one is, cause or effect, and I think we've got to realize there's a two-way process between the microbes and our food and our brain. Obviously, if you've got the right set of microbes, then you're going to be able to get rid of foods, digest them much more easily. I think that's the... So if you've got fat-busting microbes, then actually you'll be able to deal with fat pretty well. But if you don't, they might leave you bloated and having problems. But of course, the more fat you eat, then you will start to gain fat-busting microbes. In the same way that if you don't eat plants, and then you're lacking all of those fiber breaking down products. So I think there's a sense of you can build up these microbes over time that then you get adjusted to it. But if you are really changing your diet in a big way, then you could be in for a shock for a while until your body is used to it. We do know you can acquire microbes as you go along, and I think you've talked before on podcast about the seaweed story and how people can gain microbes. You're not just fixed in the amount of microbes you have. You can, just by eating lots of sushi, suddenly acquire the ability to break down some of these tougher plants, etc. So I think it's a two-way process. But so some people may have trouble eating foods because they don't have the right microbes that break them down. They then send different signals to the brain, which might say, this is difficult for me. And we do know from animal work, not human work, that microbes can send out signals to actually choose the foods for you. Mm-hmm. So, which sort of makes sense in an evolutionary way. If you think you've got the microbes that are all struggling to survive, and you've got this little microbe only likes eating burgers. If there's a way he can evolve a signal to send to your brain to say, "Oh, send me more burgers down, otherwise I'm going to die," you would do it. And we do know that in tiny little insects, just by manipulating the microbes, they can send signals out to say, eat more protein or eat more carbs. And so we know in theory it's true, but I think it's this. We definitely are a mix of our microbes, and because we're all unique, all of us have a unique set of microbes. We are going to be getting different signals, and we'll all, in a way, be able to digest food slightly differently because of that. So again, it goes back to this idea of our uniqueness and the fact we the standard diet isn't going to have the same effect on all of us.
With regards to those signals, I've always wondered this. Do you think, I know that approximately 50% of the average person's calories today are coming from ultra-processed foods. Do you think the consumption of those foods could be changing the microbiome in a way that then it's this kind of positive cycle where the microbiome is sending signals for more of that food because there are certain bacteria in there that are thriving off that?
I definitely do, yes. I think it's going to be quite hard to prove in humans, and that's why we may have to rely on animal studies to do that. Mm-hmm. But it sort of makes sense. Particularly, you see people who are on junk food diets or animals on junk food diet, they have a very pro-inflammatory set of gut microbes. So these are microbes that like an inflammatory environment. They thrive off it. And that allows them a niche because the ones that don't like it are wiped out, basically. Mm-hmm. So these guys have the place to themselves. They like it a bit hotter, if you like, than everyone else. And so it's in their interest to keep it hot, to keep the other guys away so they can dominate. And I think that's the way to look at it. So I think it's perfectly plausible that, our microbes are producing these signals to our brain to keep this whole process going. And that's why it may be difficult for some people to break this cycle or this junk food cycle, that our microbes are contributing to it as well as other bits in our brain.
You mentioned the fact that our microbiome may see us have different foods that are optimal for us, that we do best on. And I'm interested in kind of how variable you think that is across humans. Are we talking about variations of a theme of eating that is optimal for humans, or are we talking about one person who, due to their microbiome, literally will do best on a, say, carnivore or meat diet, and the next person does best on raw vegan? How much kind of personalization variation are we talking here?
Oh, it's a great question. I'm not sure I can give you any more than a wild guess at that. But it sort of depends what's your starting point, really, because I think, we all start with 0 microbes and we end up with a whole variety of different ones, and how we've acquired them might be important, what sequence we acquire them. So there's no doubt that, if you are an Inuit or, you're living in the extremes of the planet, people tend to eat more fats and more proteins than they do plants. And their genes have also adapted to be able to cope with extra fats. So I think there will be some idea that you'd expect the microbes to be very good at breaking down fats and requesting more fats, and perhaps not so good at lots of grains in those extreme bits of the planet. But I think definitely that some of this variety in our tastes could well be coming from our gut microbes. I'm sure that's true. Whether it's fixed or it's something that we can modify, I'm not so sure. there seems to be relatively three compartments of our gut microbes. One is that some change sort of every day, some hardly ever change, and some, will change over a few months if you change your diet. And I'm not quite sure how much is absolutely fixed. I have a feeling that humans are actually more flexible than we think, and that we are these amazing, successful omnivores.
Mm-hmm.
So I wouldn't like to think that everybody is absolutely fixed forever in their microbes and their tastes, and whether they're only going to eat meat or only going to eat plants. I like to think that we do have more flexibility, but it may take a long time to reach it.
Sure. It was more of a hypothetical question, of course, but I think Herman Ponce did a good job kind of explaining the wide variation in different diets in hunter-gatherers across the world. I'm sure you've come across his work. Question with regards to our microbiome, and if someone's listening and thinking, gosh, this sounds really interesting, other than kind of intuitively feeling what foods feel good and what foods maybe don't feel so good, where are we at today with regards to objectively looking at your microbiome and then getting some sort of objective feedback, information that says, Simon, these are going to be foods that perhaps due to your microbiome you're not going to do so well on, and then these other foods here you're going to do very well on them in terms of your metabolic health and how you feel?
I think we're getting along the way there, but I don't think we're at a point where we can solely use the microbiome to tell people what they should be eating. Mm-hmm. So certainly with the studies of in Zoe, we've used the microbiome along with metabolic tests in combination, looking at your, the glucose spikes and your how much residual fat you've got in your system. Alongside, there's one of as one of those three pillars, if you like, of assessing it. I think at the moment we don't quite know enough about the microbiome on its own to make these really strong predictions, but we're getting better every year. And so I can see a point in the next few years when it will, in a way, we'll have enough information about it. We probably will be able to overtake these other, purely more physiological tests. Mm-hmm. But I think to do that, we're going to look not just cross-sectionally at the moment, which is, one point in time data, but it's looking at people's microbiome over time and maybe doing interventional studies on people to say, well, if we, gave you this special diet for a month and your microbiome changed like this means this is how you should be eating. I think I see that as the logical way, but just cross-sectionally, I think at the moment, I don't think we have quite enough knowledge to be able to say exactly what you should be doing. But, things are changing very fast, and the sequencing we're doing, in 2 years, we've doubled the number of species we can identify on our metagenome analysis. So I think it's very much a question of watch this space. It's one of the fastest moving fields at the moment. So I don't want to be too restrictive. I can just say at this moment in time, I think we need to combine the microbiome findings with your blood sugar and your blood fats to come up with what's going on. But some of those things, like the blood fats, we might be able to replace with a microbiome as we're understanding which microbes are really the key fat, the fat burners, etc. So it's coming, but not quite there yet.
I'm sure there's some people listening, Tim, that are thinking this personalized nutrition, it sounds amazing, and I'm sure we're going to get there one day, and it sounds like an incredible tool for people to really optimize their diet, but we can't even get people to eat according to the dietary guidelines. And so I wonder just, how you kind of think of all of this in a big picture. Where does personalized nutrition sit in terms of addressing the chronic disease I guess, pandemic, whatever you want to call it, that we see today, where does changing the food environment and sort of other solutions sit in the grand scheme of things?
Well, I see it as a tool for changing the way everyone thinks about food. So I agree, only a small percentage of the population will be able to actually do tests like the Zoe test and test their personalised nutrition. But the fact that we talk about it, we're talking about on this show, I think is spreading the word that the old model of it's all about calories and calorie counting and basically food is fuel is complete nonsense. And, once you start realizing that everyone's personalized, there's a different response to food, and that your gut microbes are pretty key to this, you're changing the whole conversation about food. And to me, that's the most important thing, that the more people talk about this, the more we can change these disastrous ideas we have about food and the power of the food companies and lobbyists and governments, etc. So I think that's how it fits in. The more we talk about personalization, the less we talk about calories. The more we talk about the quality of food, the more we talk about people thinking about what they're putting in their mouths because it's full of chemicals and it affects all these other microbes which produce other chemicals. We're changing that whole discussion about food. We're talking about really getting into the quality of it and making people appreciate what they're eating and realizing that the 50% of food that we eat really should be taxed into the dustbin, and that's the word. So I see it's all part of this big picture, and that's what I think really engages me, gets me out of bed in the morning, makes me want to write, as spending hours and hours writing books, which is not, which never makes you any money, but it allows you to talk about this subject. And I think I'm optimistic that there's more and more people talking about this now, and it's a ground up movement. Governments aren't going to do anything. Big food isn't going to do anything. But if enough people are talking about personalized nutrition, the microbiome, they're going to be talking about food quality and the food environment. And I think that's, by combining the science with the politics, that's the way we can get things changed.
In terms of the dietary guidelines as they are today, is there a problem with them? I know that some people suggest there are large problems with the dietary guidelines. Do you see a sort of significant problem with the guidelines, or is it more the way the environment is set up that is affecting the way people are eating?
Well, the guidelines are an obstacle because they are so old-fashioned and so out of date and give the idea that everyone is identical. Mm-hmm. And they really haven't shifted much in 20 years. And they're giving people generally bad advice that most doctors still give their patients, because doctors might be one of the few people who read the guidelines. And so they're passing on this false information that, a lot of it is demonstrably false. And it stays on official websites for years and years after the evidence has changed. So that's what I don't like about it. But you could argue on a pragmatic level, well, no one only 2% of the population follow it anyway, so why does it matter? But I think but I think the fact that that's the official guidelines, when we know that if you ask most professors of nutrition around the world, they would say, well, I don't agree with half of them. We should move on. It's a problem. But I think, we need to move beyond those guidelines and just start realizing there isn't one set of guidelines any of us can write for humans, and we've had discussions about there isn't one set of calories that everyone should have. these things are rather meaningless, and actually try and narrow down our complexity of our food and our human bodies into some simple list to follow, which is either impossible to follow or is plain wrong. But it's getting us to prescribe into certain patterns, like, in my books I go into this in quite a lot, but like never skip a meal, always eat little and often, you've got to drink 8 glasses of water a day, all this sort of stuff, which is pseudoscience when you look at it now, and you say, well, why should we all do this? Why are we all the same anyway? Why don't we do what's right for the individual? So yeah, I'm against guidelines. I'm very rude about them, and I think that the only people that benefit from them is the food industry, I think is the honest answer.
If a couple of questions on that. So, if you were sitting down as a physician with a patient and you could give them 3 or 4 or 5 sort of bullet points that you felt were far more beneficial than referring someone to this dietary guidelines as they are now, what would that piece of information look like?
If I was really keeping it very simple and high level, I would say try and eat for your gut microbes. Every time you think of food, if you're eating for your gut microbes, you're not going to go far wrong. And if they say, oh, what does that mean? I'd say, well, eat as many diverse plants as you can in a week. And I say, try and go for 30. Remember that nuts and seeds and herbs and spices are all part of those plants. Try and pick plants that are colourful and have lots of flavour, because they've probably got polyphenols in it, which are rocket fuel for your microbes. Have fermented foods, which are good for your microbes, and you give a list of remind people what they are. Many Brits and Australians don't regularly have them, so you might there is this stuff called kefir and kombucha and kimchi and sauerkraut, as well as yogurt and cheese, and just say your gut microbes don't like ultra processed food much, so really try and cut that out. And then if they ask, is there anything else I could do? Well, I say, well, you could try restricted time eating, because your gut microbes like a long rest period. They like to sleep in. Give them a long time. So I think if every doctor told their patient that, I think it would help a lot more people than reciting these sort of biblical diet guidelines.
And what if they said, okay, doc, all of that makes sense. What about when I'm thirsty? Juice, smoothie, low calorie, artificially sweetened Coca-Cola. What am I drinking?
I'd say your gut microbes don't like lots of sugar, because they don't get to see any of it. So you would avoid all those things. And you should be, if you are going to drink anything, it should be water, it should be tea, it should be coffee particularly, which is now much healthier than orange juice and should be in the health food section, because it's, and you remind them that coffee actually is a fermented plant, and it's just roasted fermented plant that actually is packed with polyphenols, and actually, if you have about 3 cups a day, has a reasonable amount of fiber in it. About a third of the US fiber total comes from their coffee drinking, and so which is a frightening thought.
Is that saying that's not saying much about the average fiber intake?
No, it's fairly low. So it wouldn't go far. It wouldn't go far for the Hadza, tribe, but for the average American, probably Britain, Australian as well, it's quite a high factor. So sugary artificial drinks, avoid artificially sweetened beverages, because we don't think they're good for your microbes either. So I think you can go a long way using the microbe argument that this, do things they like, and then and that's makes you understand a bit more your body, understand more the food, what it's doing, rather than just these rules that people blindly follow, which no one really understands, even the people probably making up the rules about, why you should eat starchy foods, why have often forgotten the 30-year-old research that it was based on, which now wouldn't stand up.
So water's good. I think we can all agree on that. I need to ask about artificial sweeteners, and I know that this is probably something that you find yourself arguing with lots of people about, and I definitely don't want to incite any type of argument. I'm just aware that there probably are listeners who are exposed to different sides of this conversation and are left in the middle thinking, gosh, Tim Spector is saying artificial sweeteners might not be great for our gut bugs. And then folks like Lane Norton are saying, well, if you look at clinical trials and when they compare artificially sweetened drinks to sweetened beverages, they seem to come out better. There doesn't seem to be deleterious effects on metabolic health. How do we reconcile this? Where do you kind of land? I mean, you've made it clear where you land, but why is it that you kind of take that approach with regards to artificially sweetened beverages?
I think I came from the approach that the companies that were pushing it were really sort of pushing them as healthy drinks, and that the healthy alternative to the Cokes and the Pepsis, the Fantas, etc., and that there was really no downside to having them. They were just fun, nice, sweet things that were good for you. And so the studies that come out, initially from the Weitzman group, showing that both in mice and in humans, you could alter the gut microbiome by giving them particular artificial sweeteners, really led weight. So when I looked for the book, I went and looked at the studies about artificial sweeteners, thinking that they would definitely show that people who switched from say Cokes to Diet Cokes or vice versa would show big differences in weight when they did that. And the studies really, when you look at the meta-analysis, are very unconvincing of any clear difference between them, which is really weird because you're thinking there's a big difference in calories if people are having, switching at least 2 cans a day for something else for over a year. The meta-analysis don't show clear differences or systematic differences. You can always pick one study that does or one doesn't. But my view was that if there was a difference, it was absolutely tiny. And that despite that difference of 500 calories a day was quite amazing. So what was happening to make up for that 500 calories must be something going on metabolically. They are good for your teeth, and absolutely dentists are absolutely right to support them. You don't get tooth decay if you're using artificial sweeteners. So that is one reason to switch. But I think we do know that certain of these sweeteners are bad for your gut microbes. We don't know they all are, and they all have very different. They all work very differently on the body. Some are absorbed quickly, some go through to the gut, and et cetera, et cetera. And it's still possible they might be able to find some. We know little about stevia, for example. It could end up being okay. But again, the real studies haven't been done. But I just, my main resistance was this is a really healthy alternative to sugar, and that we should just have as much of it as we like. And we know many people that do get addicted to it, that are having 5 or 6 of these a day. And that means they're also not eating other proper foods either as a result. So that's why I'm against them. I have sometime in common. Would I rather have something I know is got sugar in it and I know it's bad for me? I know exactly what's in there, or this sort of mystery compound that has these chemicals that haven't really been tested to know what's happening. So that's why personally I'm against them. But I don't think having one diet drink a week is going to upset anybody. I think having 2 cans a day for the whole of your life is. And I think that's also an important difference.
Are there any kind of studies that you wish would be conducted with these kind of compounds? I know that you mentioned there's been some animal and human studies. I guess one thing that I do hear about animal studies, and you would be well aware of this, is that sometimes the exposure amount can be different with regards to kind of milligrams per kilogram of body mass, proportionally to what a human would be exposed to. Are there any trials that you think would help sort of shed further light on these compounds?
Well, human studies are obviously the ones to go for, or at least a combination of the rodent studies with parallel human studies. So you work out what it's doing in rodents, you get the mechanisms, and then you actually do the trial in humans. And they haven't. I think there are some big. I heard the Israelis were doing a big study. It hasn't been published yet, and they, I think, had over 1,000 people, which is the sort of numbers you need. Because of the individuality in our gut microbes, it could be that some of us are resistant to, are quite able to cope with sweeteners, and others aren't. And so I think you do need big numbers. And of course, who's going to pay for this? It's very hard to get money for proper nutrition research. And of course, the sweetener companies are doing all they can to stop this kind of research. Or they will do small, tiny rodent studies to try and disprove that there's any harm done. They don't want to do any big studies. That's totally not in their interest. So they're the kind of studies. So yeah, 1,000 people followed for a decent length of time, looking at these metabolic markers, but also looking at a range of different sweeteners, because increasingly they're used in combination as well by other companies as well. So we don't really know how they work. But I think, what shouldn't happen is new artificial sweeteners are introduced into our food chain without any tests on humans or the microbiome. I think that is just madness. So that's what I'd love to see. But I think the interesting analogy is that this work on emulsifiers in food, and there's Benoit Chassagne in Paris has been looking at this and showing that certain emulsifiers in processed foods, only some people are sensitive to, but they might be acutely sensitive to it because of their microbes. And so, there might be 6 or so common emulsifiers in processed foods, but only 2 or 3 of them may be problematic. Mm-hmm. And it may only be problematic in about a third of people. But they get which is very personalised.
Is that something that you think Zoe will be able to help someone predict in the future? So when they do their microbiome test, they're not only getting food recommendations, but they're also getting recommendations about particular additives and those that they might cope fine with versus those that they may not?
Absolutely, yes. That's very much the plan. And we're working with Benoit at the moment to try and understand which microbes are highlighted and which, so that we can do a test about likely susceptibility. So once you get your microbiome profile done by Zoe, we'll be able to say, we believe you're highly likely to be sensitive to these particular additives in food. And I'm going to start with the emulsifiers, but as you know, there are potentially hundreds of others. But it does look like that once you've got these big samples, now we've now got nearly 30,000 subjects with their diet history and their metagenomes. We're going to soon have 300,000. We'll be able to really tease apart these things, so give very precise advice, so that people who totally unknowingly are having a really bad reaction to something in food will be able to avoid them. But it probably means the manufacturers will just change it for something else, because once it gets known, a bit like, the big scare about numbers, everyone just went and they just changed the names of the chemicals to something similar.
Sure. How much of this can we potentially sort of sidestep by changing the composition of our microbiome? So I'm interested, you've got these 30,000 folks or samples in Zoe so far. Have many people submitted multiple samples over time and you've been able to see changes in the microbiome with changes in diet, and then whether that's affected blood glucose control and lipids?
So that's really the concept of retesting people, and that's we've just started analysing a pilot of about 300 people that have done it again, and we're also starting a randomised controlled trial to actually do this systematically rather than just observationally, where we give two arms, one with the Zoe program, one without, to look at these differences. But we don't have the data at the moment, but I think that's a really important question, is working out how do we focus on the modifiable parts of the gut microbiome. And at the moment, our advice is much more cross-sectional, but I think it's going to get much more concentrated and focused, because we can say, we know that in most people who go from an unhealthy to a healthy diet, these are the microbes that are crucial, and then we can work out what the foods you need to eat are for these crucial microbes. So we can start to really zone in on what we think are the key ones, because in all this complexity of the gut microbiome with thousands of species, it's different in everybody, a lot of confusion. But if you can work out what the key species are and start targeting them and getting rid of the unhealthy key species, then I think we can get much better targeted advice. So that's what we're aiming for. And I think it would be nice for people, once a year to get retested to see, well, am I on the right track? And crucially, the bit we don't know is how much, by improving your gut microbes, can you reduce your sugar peaks to the same food? So ultimately, we would like to say, after 2 years of this, you've sort of improved your good bugs by so much, you can now have extra sugar or carbs, and you won't get a worse peak. We don't know that yet. At the moment, we're telling people just to swap your foods around. But everyone has their favourite food that they would quite like to have. So knowing that in 2 years' time, it's safer to have it. And intuitively, it makes sense that if we have a healthier gut microbiome, we can give ourselves naughty treats more than if we have than if we're in an unhealthy state, because we're better able to cope with it. We've got more balance. So I'm hoping that will be the case.
On the sugar peak, I've got to actually have a question to run past you. So let's say we're talking about a healthy individual. So their HbA1c is well below 5.7. They're not pre-diabetic, and then they don't have type 2 diabetes. How important is the kind of rate at which blood sugar goes up, and the kind of trajectory of that curve for a healthy person if they're in range the entire time?
So you're saying the peak never goes very high, in other words?
Well, the peak goes up. Let's say, for example, their blood glucose goes up, but it doesn't go outside of the normal range, versus, say, after a certain food or a meal where it's also within the normal range, but it's lower. Are we automatically presuming that lower, the response to the lower meal means that lower meal is therefore healthier for them, despite the fact that in scenario A and scenario B, their blood glucose in both examples is within the normal range?
That's a good question. Well, I think we don't really know what the normal range is. They were set in the days when we were studying diabetes and pre-diabetes, rather than studying normal non-diseased individuals. So I think we're starting from scratch really to build our what is a normal range, and I see this as not a binary effect, but it's a continuous curve that in general, the more peaks you have, the more subtle inflammation or stress on the body you're getting. Mm-hmm. So that's the general concept. This idea of a normal abnormal range, I'm not sure is true. It may be very hard to see a difference at these low levels. So it could take years before you actually noticed any real effect of them. And so it may not be something to worry about at the moment. Mm-hmm. But I don't think we know yet what a normal range is, because in our studies we have shown that both glucose peaks and fat peaks are associated with low-level inflammation measured in the blood.
When you say peak, are you talking about, is that referring to a fast rise and a fast fall versus slow rise, yes. Slow fall? Right.
Yes. There's obviously, there's the area under the curve, which is the other way of seeing mm-hmm. this, of how long it does stay up, which is the sort of standard way of looking at this, and both are associated. But the actual how far the peak goes up is also linked to inflammation. So mm-hmm. I think it's going to be very difficult to work out whether there is a safe level. One level is safe because it's normal for your blood sugar to go up. You eat something, it goes up. We're born that way. We mustn't be frightened of that. I don't think we want to. To have to live a life where you have zero peaks of anything. I don't think we quite know where the level is, where there is no extra inflammation. I think maybe that's the key. To do that, we will need massive studies. We did a study of a few hundred people, which is big by nutrition standards, but it wasn't enough to come up with a safe level. I think you need thousands of people to do that mm-hmm. before you can get that space. But mustn't forget there's other things of the curve that are also important. So we discovered that this 3-hour dip. So you can have not so big a peak, but actually if you have a dip below the threshold 3 hours later, that was associated, 1 in 4 people had that, and that was associated with extra hunger. So you actually ate more during the day. And so I think we're discovering there's much more to these profiles than just a simple peak or an area under the curve. As we get more and more data, I think we will understand this. But I don't think there's such thing as a normal range. And it might be that, all of us, in a way, we all have our personal normal range. And we need to work out what that is and what is the area that changes our energy levels or makes us more hungry or makes the blood slightly more inflamed. So bigger studies will show we will get these answers. So maybe in a year I'll be able to tell you.
Do you have any sort of clues or ideas as to what might cause that dip 3 hours after a meal that then increases appetite?
I don't, to be honest. It's obviously some sort of overcorrection. Mm-hmm. And what exactly is controlling that, I don't know. I think we were the first group to look at it because no one had looked beyond 2 hours previously. And it was actually a non-medic who was doing the analysis, so he didn't know. He had a physics background, so he didn't know that you weren't allowed to look more than 2 hours. And so he looked at 3 hours and found it. So I guess it could be something in the brain, the controlling part of this complexity is mm-hmm. often somewhere in the brain that is suddenly saying this is some defense mechanism to tell us to eat more, something evolutionary, but it doesn't happen in everybody. That's really interesting. So again, we're going to be telling people, are you a dipper? And should you be extra careful about what you have for breakfast? Because otherwise you'll be super hungry at 11 a.m. And you're going to overeat in that day if you don't really watch out for these potential dips. And people will get into habits. So I think that's one of these exciting things, is how we can feed back this information so people can, and other people are maybe immune to it. They just don't dip. They don't get hungry. And I think that's also really interesting. Why are they protected? Why don't they get it?
Those that do experience that, do you kind of hypothesize off other research that they may do better with a higher protein, higher fat meal, less carbs, to try and avoid that dip? Or what do you think you'll find?
Yeah, I think those people generally do better upping their fats and having less, fast available carbs, because it's quite a big factor. If you just had a carb breakfast, for 10 years, and you're putting on an extra, in the short-term study, we showed these people were putting on, 250 calories more. That's going to be a considerable weight gain that they wouldn't notice, and they wouldn't know why they were just eating more. And again, I think it's a brilliant study to show how ridiculous the idea of calories is, because everyone was eating identical breakfasts, and yet some people, it made them overeat in other meals later in the day, and others it didn't. And I think that one experiment really brings home to me the power of personalized nutrition and advice, and blows out of the water the idea that, all calories are equal, and that, everyone should have their 2,000 or 2,500 calories.
You mentioned before continuous blood lipids, or you mentioned that you were measuring blood lipids, and it makes me think about, is there a future where, at the moment we have continuous blood glucose monitors, do you think that there would be benefit from monitoring lipids continuously? And I'm interested in sort of why you added that lipid test within the Zoe framework. Why is that important?
Well, we added it because we realized that, if you could, for some people, fats are a problem. We've known that fats hang around in some people's blood for a long time, and they're associated with inflammation. And, fats have been demonized for a long time, particularly saturated fats, because of the pro-inflammatory effect they have and the fact they increase heart disease and diabetes. But we've known there's this big variation. And when we were setting up Zoe, obviously the CGM, the glucose monitor, was there, but we felt that you can cheat on your glucose monitor. You can say, okay, if you just add double cream or to everything you eat, you can just about get a flat line, right? So, are you going to say to everyone, just with every meal you have a tub of double cream as fat, and you can have what appears to be a perfectly healthy diet? And most would say, well, probably that's not a great idea. So, in a way, the idea that we had to wanted a more holistic approach, rather than just be obsessed purely with controlling the peak and carbs. We wanted to look at other aspects and realize there were people who might be poor metabolizers of fat, that if you switched them from a carb diet to a high fat diet, would have these residual bits of fat in the blood, that we know that if at 6 hours you've still got a considerable amount of fat floating around in your blood vessels, that breaks down into these small little particles. And these small particles get caught up in the lining of the blood vessels and cause inflammation and atherosclerosis and all kinds of other problems and inflammation. So we wanted to be able to give advice both ways, to say, okay, we think you're not coping very well with carbs, and luckily, you can have as much fats as you like, or you can do this, but you can only go up to a certain amount of fat before you might start problems. You need to be not eat it too much in one go, because your body may not control it right. And that was the rationale. And we'd love someone to invent a continuous triglyceride monitor, and we probably wouldn't need it every 5 minutes, but you could at least have it every hour. It hasn't been invented yet, but if someone's listening, I think you'd make a fortune if you can invent one. Because look at the billions that the glucose monitors have made for those companies. But at the moment, we're just doing it with a finger prick test, and there is quite good technology that does it on a dry blood spot, and so it can be done in your home. So these have been really big advances about measuring triglyceride at home very, very easily and cheaply. So that's why it's this holistic approach. I don't think you can just take personalized nutrition and say it's only about sugar peaks or it's only about fats. But if you combine them and then you add in the mix of the microbiome, then I think you're getting much more towards a holistic view of nutrition, which is where I think we should all be heading.
Yeah, I think that's really sensible, and it kind of speaks to where I was going earlier about the peaks. I have seen some communities online where the CGMs are very popular, that some of the rhetoric is about flatter the better, and one of my worries was that, like you just said, you can kind of hack your way to a flat blood glucose, but is that negatively affecting your diet quality in that process and having a deleterious effect on lipids? And you wouldn't know that unless you're actually measuring them, like you've just explained. So I think that's great. Tim, there's so much more I want to talk about. I'm just conscious of your time and your schedule today. I want to get you back on and talk about dairy. I want to talk about fish. I want to talk about snacking, meal timing. There's a lot more to delve into, but I know that you're very busy and I want to let you go today. So thank you so much for what's been a very, very informative discussion. I really appreciate all the work that you're doing. As I said at the outset, I've read a lot of your published papers. I've certainly read your books, and I think you are super insightful and a great voice in this space. Leave us with a bit of a look into the future. Let's say it's 2032, so a decade from now. Time will fly. We'll be there before we know it. Have we made any inroads as a society with regards to public health and nutrition in that 10-year period? And what does personalised nutrition look like then?
Wow. Well, I hope we've cleared COVID by then.
Me too.
It'll be COVID Mark II.
Yeah.
We'll have the new virus, the new Aussie virus for this. But so I think personalized nutrition will be with us. I think we'll be wearing smartwatches that can measure our glucose. We'll be getting sugar readings through our watches, and we'll be much more aware of the effects of foods going into our bodies. Hopefully, it'll also measure fats by then as well, so we'll know that. So it'll be a consumer-led approach to foods, and I think food labeling will have changed, and hopefully it might be a Zoe label on foods telling us what the microbiome score is for that food. That says how gut friendly this food is, which is will be a measure of quality. I think calories will still be there, but hopefully in a minute font, where they as small as they put the fiber font on the labels now will be where the calories are. And I think the food companies will have adapted and changed. There'll still be ultra processed foods, but they will be forced to study those foods and those additives for their effect on our gut microbes and our general health. So I can't see that. I still think 50% of our food will be ultra processed. That's not going to change, but I think all those companies will have shifted to have more gut-friendly products by then, because they already have divisions within those companies working on this. And also personalised nutrition will be a real thing. So all the products will have different colour coding for saying whether you're a good or bad sugar metabolizer or fat metabolizer, etc. So I'm an optimist. So I think there will be this opinion, this group of products. It's not going to be universal because it may, even in 10 years, I don't think everyone will be able to afford these level of foods and devices. But I think it will be very much mainstream, and everyone will be testing their microbes every year, definitely. This will be, and all the health services will do that. Rather than measuring your blood cholesterol, this will be a much more important thing for all of us to do. So we can talk in 10 years and see how wrong I was.
Yeah.
All right.
I'll hold you to that. And of course, a number of other things that I'd love to get you back on. I just mentioned a few there, but I'd love to dive deeper into ultra processed foods and specifics with regards to how they could be improved and your thoughts on that. But we'll save that for part 2. We'll keep this as an open conversation. Thank you so much again. Look forward to having you back on. I'll pop a link to your socials, the Zoe website and their socials, and also to your books in the show notes.
That's great. Great talking to you, Simon.
Thanks, Tim. Thank you for joining me for this episode and your interest in science-based conversation. I hope you enjoyed it and found the information covered interesting and instructive. If you did and you'd like to show your support for the show, please subscribe to our YouTube channel where you can stay up to date with new episodes and watch them in video format. Yes, the full-length videos. Please also consider subscribing to the show on the Spotify and/or Apple Podcast app, wherever you enjoy listening to podcasts. You can also leave a review on Apple or Spotify. Again, a great way to support the show and make our content more discoverable for others to enjoy and learn from. If you have any comments about the episodes, suggestions for future episodes, including guests you'd like to see on the show, or questions that you'd like to have answered, please leave those in the comments section on YouTube. I myself and my team will take note of these comments when planning future episodes. Finally, the best way to support the show and receive discounts on products we love is by checking out our sponsors at theproof.com/friends. Enjoy your week, stay well, and I look forward to catching you in the next episode.